Dear student, thank you for sticking with me. You're gonna be hearing now about a new topic in this module, circulation. This is mini lecture number 4 and have to do with shunts and the fetal circulation. Essentially, if we revise, we start by looking at the adult circulation in a mammal or in a bird. What this circulation is characterized is by the existence of two parallel circuits that are at the same time of the non-mixing nature. And that means that we have a circulation towards the lungs, from the heart to and from the lungs, and from the heart to and from the rest of the tissues in the body. That's what we call the pulmonary circuit and the systemic circuit. The systemic circuit is what provides perfusion to the brain, to the gut, to the intestine, to the heart, to the legs, to all muscles essentially, versus the pulmonary that provides the circulation to the lungs. So, two non-mixing parallel circuits, meaning that a red blood cell coming outside of the right ventricle will first go into the lungs, return to the left atrium, be pumped to the rest of the body from the left ventricle, and then eventually come back to the right atrium to go back into the lungs again. These are two parallel circuits that are not mixing with each other. But of course, we all mammals, all birds, go through a developmental stage as a fetus in which the lungs are non-functional. How is this fetus obtaining oxygen? And essentially there are a set of structures that allow that. In mammals it's the placenta. In birds it's the chorioallantoic circulation in the egg. Both chorioallantoic, the placenta, are connected to the systemic circuit. Meaning that as a developing fetus, the structure of the adult cardiovascular system is useless. It's useless in the sense that because oxygen cannot be obtained in the lungs, we need to make sure that blood can cross both sides and still maintain a viable circulation while the animal is developing. And of course that means that there is going to be an important point in life in the transition from the uterine or the egg life into a single standing organism, a terrestrial one, basically, in which the circulatory patterns will have to change and to become the typical adult cardiovascular system or the typical adult heart circulation. And this is basically the representation of a fetal heart. That, for the most, it's very similar to the adult heart. What you see is two atria, two ventricles, and the outflow tracts, the aorta, the pulmonary arteries, and the returning into the heart, essentially the same. But there are two particular features that make this fetal heart unique, and that allow this fetal heart to function during fetal life, to oxygenate the blood from the placenta and continue its suitable and proper development. And if you pay special attention here, you will see something that looks a bit strange. And what looks strange in this circulation is, first, that between the pulmonary circulation and the systemic circulation in the aorta, there is a connection. This connection is not present in the adult heart. This connection is called the ductus arteriosus. The second element that differs here is the connection in between the atria. The right and the left atria are connected with what's called the foramen ovale. And these are the two structures. These are the two structures that distinguish the fetal circulation from the adult circulation. And these structures actually are the origin of most of the congenital heart disorders that we see in newborn babies. The question is why? Well, pay attention, and this is what's important here, is that this ductus arteriosus in fetal life is important. Well, both actually, the ductus arteriosus and the foramen ovale, are important to discharge or to relieve the pressure that the right atrium would get otherwise, considering that most of the blood goes into the systemic circulation and relatively little go into the lungs that are non-functional, that are still developing. The ductus arteriosus will take an important portion of the pulmonary artery flow. So the blood flow in the pulmonary artery will split up part of it, up to 60% of it, into the aorta, and the remaining will go to the lungs. Notice that by default puts a lot more blood in the systemic circuit, that if there was no connection between the right atrium and the left atrium, would mean that would have to be pumped by the right ventricle to release that excessive flow returning into the right side of the heart. That is the foramen ovale that takes part of this flow through it. So both the foramen ovale and ductus arteriosus are fully functional in a fetus, are extremely important in terms of allowing fetal development as it proceeds. But something has to happen with these two structures at the time of birth. Improper closure of these two structures will mean simply that oxygenated blood or deoxygenated blood will be mixed up with the other circuit. And that's where the concept of a shunt appears. A shunt is an alternative pathway that diverts blood flow from one major route, the systemic or the pulmonary, to the other. And this is actually the cause for what we call blue babies. Blue babies are babies that look bluish because the oxygen saturation in their blood is lower due to mixing of oxygenated and deoxygenated blood at the level of the heart or at the level of the circulation, because some of these fetal structures have not closed properly. This is essentially a very cartoon-like representation of it, but the right atrium will feed the lung circuit, the left atrium through the left ventricle will feed the systemic circuit, and in an adult scenario there should be no mixing. But we already have seen that there is a mixing in the forms of these two fetal structures. And these two fetal structures, the foramen ovale and the ductus arteriosus, have to close at one point. If they don't close, it could result in oxygenated blood moving into the lung circuit. Therefore, we are sending oxygenated blood back to the lungs for no purpose. Or deoxygenated blood, instead of going to the lungs to become oxygenated, going into the systemic circuit, with the result that the blood that is distributed in the aorta is simply less saturated with oxygen. Of course, by definitions, there are no shunts if there is no parallel circulation. So if we look at vertebrates as a whole group, we cannot have shunts in water-breathing fish. Shunts can be intracardiac and extracardiac. In this case, for example, the foramen ovale is an intracardiac shunt path. The ductus arteriosus is extracardiac because it's found outside of the heart. And then cardiac shunts can be either right to left, that means blood from the right is going to the left. The blood that comes back to the right side of the circulation is deoxygenated blood. So we are sending deoxygenated blood back in the systemic circuit. The final result is decreased oxygenation of the systemic blood because we are bypassing the lungs. Or the left to right shunt, in which systemic blood, oxygenated blood, is sent back again to the lungs and we are recirculating pulmonary venous blood. These are the kind of shunts that we can see. Right to left shunts reduce oxygenation, and left to right increase the oxygenation, but one can argue if this is helpful if the blood is already fully saturated. The conventional shunt model is what I showed you earlier, and basically this would be a right to left, and this would be a left to right shunt. We're going to see now for the rest of the mini lecture how this affects babies, and then some evolutionary implications that this will have for next mini lecture in this module. Shunts are an essential feature, as I said, of the embryonic and the fetal circulation. They are necessary for things to work and for the cardiovascular system to mature appropriately. But they are also the typical, it's actually what causes, it's what is at the base of most congenital heart defects. And that means that the failure of closure or malformations around that time will be seen at the time of birth, typically in the form of blue babies. This is one of the most severe malformations of congenital defect, and I just explain it as a sort of an example. This is what we call the tetralogy of Fallot, and that's because it was discovered by a French doctor, Fallot, and it consists of 4 different dysfunctions or 4 different abnormalities or malformations. Number 1 is a VSD, a ventricular septal defect. That means that there is no closure in the septum between both ventricles. And that, of course, is an avenue for possible exchange of oxygen-degenerated blood. Second aspect is a pulmonary stenosis. That means a narrowing of the pulmonary artery. Narrowing of the pulmonary artery will increase the resistance in the pulmonary artery and will basically decrease the amount of blood flow going into the pulmonary artery. If it can be shuffled, it can be shunted into a different direction. In this case, through the VSD, the ventricular septal defect. Hypertrophy of the right ventricle, that means that the wall of the right ventricle thickens as a result of the increasing pressure because of the stenosis. So this right ventricle is working more, and you have a thickened wall of the right ventricle. Usually the right ventricle is thinner than the left. In this case you see they are equal or even thicker. And finally the problem of an overriding aorta, in which the aorta position and the valves basically can take some blood from the right ventricle as well. This is a very severe effect that can be corrected, is corrected typically even now intrauterine, when it's the best time for doing that. There are many other congenital heart defects. A VSD in itself, only a ventricular septal defect, the tetralogy that I just talked about, or many of the defects seen in the tetralogy can be also found by themselves. Stenosis of the pulmonary valve, an atrial septal defect. Instead of this defect in the septum, in the ventricles are actually seen in the atrium, and that is because of an improper closure of the foramen ovale. Hypoplastic left heart, in which the left ventricle grows too little and is reduced so it cannot function properly. A patent ductus arteriosus, that means that the ductus arteriosus does not fully close after birth, or even an aortic stenosis. So these are all different congenital heart defects that will have an important effect on cardiovascular function in the baby after birth. Most of these defects may not be important during intrauterine life, but then they become severe after birth, when it's important and proper to have a double non-mixing parallel circulation. The most common heart birth defect is actually the VSD. About 1 in 500 babies are born with it. And about 30% of all heart defects are actually ventricular septal defects. So it's not uncommon. And in many cases, if the defect is small, they can go unnoticed for years. And that's because the small VSDs do not require treatment. In many cases, they may close spontaneously on their own. In large VSDs, though, in large ventricular septal defects, what happens? What we see in large ventricular septal defects is a large LR shunt. So that means that blood from the left is shunted into the right. Why is that? That happens because the right circulation is a lower circulation circuit. So of course, based on a pressure gradients, the tendency will be for blood to leave. If there is an opening between both ventricles, the higher pressure circuit will pump into the lower pressure circuit. So that is and left to right shunt. And this left to right shunt will recirculate blood through the lungs. And in terms of oxygenation, that may not be a gigantic problem. But the problem, of course, is that it reduces cardiac efficiency. The heart is working for no reason. The heart is pumping blood back into the lungs when it's not needed. There's no need for oxygenating it further. So, of course, ventricular septal defects, if they are particularly large, will have a severe impact on cardiac function. This lung recirculation that I'm talking about with the VSD will lead also to pulmonary hypertension. All of a sudden, there is more blood flow going into the lungs. There will be an increase in resistance, an increase in pressure. Eventually, if the heart has to work excessively, can lead to heart failure. Large ventricular septal defects are corrected, should be corrected. The earlier the better. If it can be done intrauterine, better than it's done later on. And how they are corrected? Simply by patching it, patching the hole with synthetic material that actually prevents the mixing from occurring. Obviously, this brings the picture that in adult mammals, shunts are maladaptive. Shunts are a problem. Shunts lead to the mixing of oxy and deoxygenated blood, and that will trigger cyanotic congenital heart disease, meaning basically blue babies, meaning poor oxygenation of the blood, appearance, bluish appearance, because the blood is not fully saturated with oxygen. That's what's seen here. Cyanosis of the nail beds, for example. This, you see again, in this case, a normal heart. If we portray this normal heart, you see the right ventricle, the left ventricle in red and in blue, indicating deoxygenated, oxygenated blood. The aorta. Notice that if the ductus arteriosus is patent, what happens here? That would be another problem. That would be another demonstration of this shunt. In this case, what you have is the same problem as I showed you earlier for the ventricular septal defect, the same VSD, but when the ductus arteriosus has not closed properly. Because of the higher pressure in the systemic circuit, blood is now, oxygenated blood is shunted away back into the lungs. Same problem, different origin, but same basically functional consequence. So the point is that these shunts are not the shunts are, I put the word maladaptive here, meaning that they are not convenient. They lead to a decreased function. But actually, if we look at the structure of the heart across the vertebrates, what we will see is that several groups in which, before groups of animals in which the heart has not fully separated, these animals are fully functional, and they actually have survived for millions of years with such a, what we call in mammals, a poor design. Looking at this, obviously what we see is that the shunts appear when there is a possibility for a double circulation, and essentially that means the appearance of the lungs as respiratory structures in terrestrial animals. Essentially, in a phylogenetic context, this would have appeared at the point of the diversification from fishes to lungfishes and the rest of the tetrapods, amphibians, reptiles, birds and mammals. And it's at this point where shunts can become, obviously, historically, phylogenetically, there was a situation in time in which the ventricles were not divided until this division occurs in the mammals. There's been a question in the literature about the possibility that these shunts that are obviously maladaptive, that obviously are not convenient in mammals, could serve a function, could have a function in other groups of vertebrates, particularly reptiles. And this is something that is appealing. It has been extensively studied. Obviously, if we look just briefly at the cardiac structure, at the circulatory patterns in some of the other vertebrate groups, we will see lots of features that remind us of the archetypical structure in the mammals, but some other features that we will consider poorly evolved or poorly designed. Nevertheless, these animals do fine in the environment. We take the amphibians in this case, or even lungfishes, that actually in particular amphibians can use the skin as a gas exchange organ. Amphibians actually will have a blood vessel leaving the heart called the pulmonocutaneous artery, in which blood is sent to the lungs but to the skin as well. So these animals can actually oxygenate blood using one of the two alternative exchange organs. The amphibian heart has two atria but only one ventricle. But what's particular about it, as we will see in the next slide, is this ventricle, the outlet of the ventricle is actually contains a spiral valve that apparently is quite efficient at separating oxygenated and deoxygenated blood. So obviously the presence of one single undivided ventricle does not mean that blood mixes fully and totally. And that's what I write here. Despite the presence of anatomical shunting structures, there is a good separation between oxygenated and deoxygenated blood. Let's take a quick look at this in the heart of the amphibians. And what you see in this heart are the two atria, the right and the left, and then a single ventricle. But you notice that as this single ventricle pumps out, it pumps into the aorta, which is a very sophisticated blood vessel that contains a spiral valve. The spiral valve, you see the flap of the valve like this here. And as indicated by the arrows, this spiral valve seems to be quite efficient at keeping the deoxygenated blood shown in blue separate from the oxygenated blood shown in red. And the deoxygenated blood in blue actually is sent towards the pulmonocutaneous arteries, while the oxygenated blood is sent towards the systemic arteries. So, the existence of a single ventricle does not necessarily mean a full separation of oxygenated and deoxygenated blood. In fact, the system is particularly efficient. And what you see here is the degree of mixing, but in red we always indicate oxygenated blood, blood returning from the lungs of the skin, returning either to the left or the right atrium. But notice that because of the presence of the spiral valve, most of the blood, oxygenated blood returning from the skin into the right atrium, will end up in the systemic circuit again. And it's the deoxygenated that will go into the pulmonocutaneous artery. This is interesting and this is relevant because these animals with a single non-divided ventricle are still able to do fairly good in separating oxygenated and deoxygenated blood. Obviously, this is not the pattern that mammals chose to employ. In mammals, there's been a completely different, a complete separation. And actually, this separation occurred as early or as late as the crocodiles. In crocodiles, we have a full separation, as we will see in the next slide. So that means that one thing is to talk about shunts in mammals, maladaptive. The other is the degree to which blood mixing can occur, even if the ventricles are not fully separated, as we would expect as the optimal solution in mammals or even birds. A last slide that basically will prime you, hopefully, to watch the next mini-lecture here, in which I will be talking about divided and non-divided reptilian hearts, is this. And that has to do with the presence of anatomical and functional shunts in reptiles. Reptiles are a particularly interesting group in which the anatomy of the heart varies quite profoundly, from turtles that have a fully non-divided ventricle in which the degree of shunting is rather severe, all the way to the crocodiles that actually have a truly separated and divided heart. In crocodiles, as you see in this graph, there is a full separation between the left and the right side of the heart. There are other avenues for shunting in other locations. You see particularly here, and I will discuss it in the next lecture, but the heart is fully divided. Versus the situation in turtles, but also lizards and snakes, in which the separation is possible, but as I will present to you later, different species solve this in a different way. Some lizards and snakes in particular are able to fully separate oxygenated blood despite the fact that their ventricles are not truly divided. And that's a particular nice story that actually goes along well with why in mammals shunts are maladaptive and not convenient, but why in reptiles or even amphibians these things have been solved in a different way and do not seem to pose a particular problem. I'll leave it here for the time. Thank you very much for listening, and I hope you enjoyed this.